그대로 남겨놓고 예전에 우리가 추노에서 본 것처럼 이마에다 낙인을 찍듯이 논문에다 낙인을 찍어요. 그래서 리트랙티드 아티클이라고 해가지고 이 논문은 표절된 논문이다 라고 해서 이런 식으로 아주 낙인을 찍어놓죠. 그런데 문제는 이런 낙인은 요즘 기술이 하도 발달해가지고 이제 뭐 100년 전의 논문까지 볼 수가 있거든요. 이 논문이 처리가 됐다고 해서 우리나라처럼 뭐 살인죄도 공소시효가 한 15년이잖아요. 그런데 이건 평생 가요. 평생 가는 의미는 여러분들의 아들, 딸이 나중에 학생이 돼서 아빠가 예전에 무슨 뭐 교수셨는데 어떤 분야에 과연 어떤 논문을 쓰셨을까 라고 검색을 했을 때 이런 그 낙인이 찍힌 논문이 나온다는 얘기입니다. 너무 과도한 처벌 아니냐. 예를 들어서 살인죄도 15년이면 죄를 면해주는데 겨우 이 논문 표절이 하나 있다고. 그런데 이거는 그 도덕성을 최우선으로 하는 이런 사이언스 커뮤니티에서는 이거는 씻을 수 없는 범죄로 절대 일어나서는 안 되는 범죄로 간주하기 때문에 이런 식으로 퍼니시먼트를 강하는 겁니다. 그런데 과연 이런 논문은 어떻게 발견되는 걸까? 논문 심사 과정에서 단 1초 만에 딱 드러날 수 있어요. 왜냐하면 요즘 같은 경우에 논문 심사를 할 때 표절 프로그램을 돌립니다. 리뷰어들이. 그럼 컴퓨터가 그때 몇 % 일치를 하나씩 보여줘요. 아니면은 지금 당장 그런 어떤 뭐 이미지를 예를 들어서 얼마 전에 그 서울대학교 그 약대의 강수경 교수님처럼 텍스트가 아니라 이미지를 변조해가지고 하는 경우도 있거든요. 이미지는 아직까지 디텍팅하는 시스템이 없습니다. 그런데 우리가 일상생활에서 뭐 그 얼굴 인식, 영화에서 자주 나오잖아요. 홍채 인식도 아직까지는 그 큰 수요가 없어서 이미지 검사까지는 안 하고 있는 건데 이미지 검사까지도 언젠간 될 겁니다. 그러면은 나중에 언젠가 시스템을 보였을 때 그 논문이 표절로 밝혀지게 되겠죠. 논문이 출판이 된 다음에 10년이 지나서 표절 안 걸렸으니까 표절 아니다가 아니라 발견되는 시점에 그 논문을 처리하게 되는 거기 때문에 논문을 한번 표절하면은 언젠간 걸린다라고 생각하시면 됩니다. 이게 그 시스템에 의해서 디텍션 되는 하나의 페이지 샷인데요. 예를 들어서 크로스 책이라고 해서 요즘 모든 논문이 등록이 되는 어떤 사이트가 있습니다. 그래서 A란 논문하고 B란 논문하고 유사도가 얼마나 되는지. 그래서 보면 지금 여기서 보시면은 has emerged demonstrate 다 똑같이 나오죠. 그러니까 일치가 되는 건 이런 식으로 이제 표시가 되게 되는 거고요. 그런데 이게 논문에서만 그 비교 대상을 삼느냐? 일반 인터넷에 있는 블로그에 떠있는 글이라든지 그런 그 구글에서 검색되는 그 아티클, 그 일반적인 그 글 같은 것이랑도 비교를 하게 됩니다. 그래서 논문량만 비교하는 게 아니라 그 웹에 올린 뭐 에세이라든가 이런 것까지도 다 표절 검사를 하게 되는 거고요. 이런 식으로 피어 리뷰 과정에서 하기 때문에 우리가 알고 있는 표절이 된 문제는 이 과정을 요행이 어떻게 잘 빠져나간 건데 나중에 뒤늦게 들킨 거고요. 그럼 이제 그 피어 리뷰 과정을 잘 지났다고 끝나느냐? 절대 그렇지 않습니다. 왜냐? 그 여러분들의 동료가 한번 앙심을 부었다. 예를 들어서 제가 나보다 너무 잘 나가는데? 아니면 제가 나한테 어제 술 안 마셨어. 이런 식으로 해가지고 가장 여러분의 연구를 잘 알고 있는 사람은 교수님보다도 여러분들의 동료가 될 가능성이 있습니다. 예를 들어서 지금 강수경 교수님 논문 같은 경우에요. 아직 이거는 그 표절이라고 확인된 건 아닙니다. 아직 그 논문 심사 중에 있고요. 그 연구 진실성 센터에서 나중에 이제 조만간 그 결정을 하게 되는데 제가 말씀드린 건 이분이 표절했다고 말씀을 드린 게 아니라 이분의 논문이 왜 문제가 됐냐는 그 절차에 대해서 설명을 드립니다. 그래서 보시면은 익명의 국내 제보자가 파워포인트로 제보를 했다라고 나오죠. 그럼 한번 그 파워포인트는 어떤 식으로 제보를 했나 볼까요? 예, 이게 그 제보자가 보낸 그 파워포인트 원본입니다. 강수경이라는 파일 제목이에요. 자, 이 다음의 페이지는 뭐 조작된 페이지가 있다. 어떻게 이렇게 그 윤리적으로 심각한 사건을 가만둘 수가 있느냐? 내가 이런 그 서울대학교 연구진실성센터 소장 이메일과 뭐 그 연구재단 소장 이메일 그리고 뭐 뭐 관련된 사람의 소장 이메일을 보내서 이분가 이쪽으로 빨리 연락을 해서 이번에 논문을 빨리 철회시켜라라고 고발하는 내용입니다. 그래서 그 고발하는 내용까지 이렇게 요약을 해놨고요. 그래서 키포인트, 뭐가 가장 문제다. 그리고 뭐 어떤 문제가 있었고 스스로 질문까지 하죠. 이렇게 이런 저자롭게 가만둘 수가 있느냐. 그러니까 이건 뭐 격가지 얘기인데 보니까 한국인이에요. 보니까 돈 스탑 버스 저자를 스탑을 해라. 통보를 시켰습니다. 그럼 보시면은 뭐 이런 식으로 논문이 이렇게 바뀌었다. 그리고 뭐 저널 요거에는 예전에 썼던 이미지가 이런 식으로 똑같이 베낀 거다. 라는 식으로 이렇게 70페이지가 넘는 자세한 얘기까지 또 보냅니다. 그러면은 이제 이 논문은 철회가 되게 되는 거죠. 거의 대부분. 아직 결정을 안 났으니까요. 그렇기 때문에 말씀드리려고 하는 거는 이제 언젠가는 잡힌다. 라는 이제 설명을 시켰던 거고요. 그러니까 뭐 제가 드린 말씀은 표정을 하지 마십시오. 언젠가는 그게 발각이 될 겁니다. 네, 제가 맡은 그 40분은 지금 시간이 났고요. 혹시 질문이 있으신가요? 네.
저기
뒤에 마이크.
네, 마이크 드리겠습니다.
예. 강의 잘 봤습니다. 근데 아까 방금 같은 케이스에서 기존에 뭐 발표를 하다가 이제 연구원들도 많이 진행된 상태에서 발표를 했는데 실제로 이제 저널에 낼 때는 그 결과가 조금 수정 보완돼가지고 결과가 조금 바뀔 수도 있지 않습니까? 그에 대한 경우에도 비교가 되는 건지.
지금 잘 말이 정확히 안 들리는데요.
예. 그 연구 결과가 어느 정도 많이 이제 진행된 상태에서 어디 뭐 학술대회나 그렇게 발표를 해가지고
네.
이 PPT나 이렇게 자료가 있는데
네.
나중에 이제 이 혹시 끝나고 난 다음에 조금 이제 연구가 수정이 조금 보완돼가지고 결과가 조금 발표 당시와 조금 다르게 나와가지고
네.
저널에 실릴 수가 있잖아요.
네.
그런 경우일 수도 있는데 방금 같은
그런데 예를 들어서
케이스는
요약을 하자면은
예.
프로시딩에서 일단 어떤 학회에서 발표를 했는데
네.
발표를 했을 당시에는 좀 준비가 덜 돼가지고 나중에 좀 더 보완해서 다시 논문으로 투고를 했을 때 이중계제가 되는 건 아니냐?
아니, 아까 방금 같은 케이스에서 그렇게 고발을 하게 되면 그런 것도 집어내는 건지.
예. 예를 들어서 그 학회가 어떤 학회냐에 따라서 그게 좀 달라지는데요. 국제학회다. 그러면은 국제학회 같은 경우에 대부분 다 그거를 프로시딩 올려놓거든요. 그럼 디텍션이 됩니다. 왜냐면 인터넷에 떠도는 글까지도 세계를 하기 때문에. 그런데 우리나라 한글로 된 뭐 누구든지 관심을 안 갖는 학회에서 잠깐 발표를 했던 거다. 그런 거라면은 일단은 그 시스템이 알 수는 없겠죠. 그런데 그것도 나중에 기술이 발달을 해서 그것도 이제 색인이 됐을 때 그럴 때는 시스템이 먼저 걸러서 뭐 표절 가능성이 있다라고 그 얼러팅을 해줄 수는 있습니다. 그러니까 그럴 경우에 차라리 좀 더 개선된 논문을 쓰실 때 사이테이션을 분명하게 하세요. 아니면은 논문 투고할 때 내 논문은 언제 개최된 어떤 행사에서 프로시딩으로 발표된 자료인데 보완을 해서 다시 제출한 자료다. 이 심사를 하실 때 참고를 하시라. 라고 하면은 그 심사자들이 그걸 보고 게재를 할지 말지 결정을 하시게 되겠죠. 네.
강의 잘 들었고요. 출판 업계에서 일하시는 분들은 대체로 전공이 이공계 쪽이 많은가요? 아니면 다른 인문학 쪽에서 배우고 오신 분들이 많으신가요?
일반적으로 볼 때는 전공 상관은 별로 없습니다. 그냥 저 같은 경우도 지금 그 인문사회학 쪽으로 나왔거든요. 문헌정보학과. 그래서 충남대학교에서 정보학을 했고요. 그런데 우리 에디터분들을 보시면은 에디터분들은 다 그 과학기술 쪽의 백그라운드를 가지고 계세요. 물리학, 아까 제가 소개시켜드린 그 원더우먼 같은 경우에도 라이프 사이언스 쪽으로 그 전공을 하셨고요. 네, 그래서 이쪽 에디터분들은 다 과학기술 쪽에 백그라운드 가지고 있고 뭐 영업이라든지 마케팅이라든지 그런 경우는 그쪽 그 디비전 성격에 맞는 백그라운드가 가지신 분을 저희가 선호합니다. 저희가 그렇지 않아도 직원 하나 뽑고 있는 거예요. 예. 예, 그러면은 마치겠고요. 그 모든 세션 끝나고 다시 종합적인 또 Q&A 세션이 다시 있을 겁니다. 그럼 이제 그 이번 이제 논문 작성법에 대해서 말씀을 해주실 분이 닥터 와렌이시는 분인데요. 이분을 간단히 설명드리면은 아까 방금 말씀하신 그 취지랑 비슷하게 실제로 논문을 써보신 분입니다. 박사학위를 소집하셨고요. 그리고 그 연구도 하신 분이고 이 아래 보이신 학교에서 강의도 하신 분입니다. 이 에단지라는 그룹은 논문을 이제 투고를 하기 전부터 컨설팅을 받을 수도 있어요. 도대체 내 논문은 어떤 저널에서 실어야 되는 거냐, 어떤 식으로 방향을 잡았는데, 이 논문은 어떤 식으로 하면 되겠다. 그 논문을 딱 투고를 했을 때, 그럴 경우에 저널마다 그 요구를 하는 레이아웃이 다릅니다. 그 레퍼런스 베이스도 다르고요. 그런 것들을 다 맞춰서 에디팅도 해주고, 내가 좀 영어가 좀 부족하다, 그럴 때는 랭귀지까지 해주는 그런 전문적인 회사고요. 그래서 이분이 여러분들이 궁금해하는 논문 작성법에 대해서 많은 지식을 가지신 분입니다. 그래서 그 닥터 와렌을 여러분께 소개해드리겠습니다.
Okay, thank you very much, JB. And I'd like to thank Kaister and Springer for organising this event today. Before I start, I just want to make you aware that the presentation today is available for download right now. What I don't want you to do is copy down all of the slides as I present them. Instead, make notes on the things that I say that aren't written on the slides. So this will be available well after today's presentation. So you can get the whole presentation and review everything I've talked about today. So I see a lot of students in the audience today. How many Masters and PhD students in the audience? Put up your hands, please. Okay, are there any staff members here? A couple of staff members. How many people here are studying in the physical sciences? Chemistry, physical chemistry, engineering, physics? What about life sciences, biomedical sciences, molecular microbiology, immunology? Okay, so we've got a good mix of people here today. So as JP said, I'm here to talk about publishing. I'm here to help you with writing manuscripts. Not just manuscripts for journals, but manuscripts for books, helping you write your thesis, helping you get published, helping to speed up the publication procedure. I'm the Senior Life Sciences Editor at Edanz Group. We're a scientific language editing company based in Japan. And I want to give you as much advice today and help you out as much as possible when it comes to publishing. Now, JB's already given some background about me. I've got a PhD in molecular virology from Murdoch University in Western Australia. And I've worked in various life sciences fields, and I've been an author, first author, co-author on many peer-reviewed publications. I've also edited hundreds and hundreds of manuscripts written by authors whose first language isn't English. So I'm very familiar with the problems that people face when it comes to publishing when English isn't their first language. And I've given many workshops like these around the world. So I've broken up today's presentation into four sections. background on scientific publishing, things to think about before you start, how you go about structuring a manuscript, and some practical hints and tips that you can use when it comes to writing. So the first section is our background about publishing, scientific publishing. Now, as researchers, we have to ask ourselves, why do we publish? Why is it important to publish? Well, we all know that the world around us is very complex and that we need to use very complicated, very complex technologies and methods to try and understand the various phenomena that we see. And as it almost always turns out, the science that we need to explain this is often very, very complex as well. And so you as up-and-coming researchers, as emerging researchers, you need to exchange your ideas with other scientists around the world. You need to do this so that people don't unnecessarily repeat your work, but also to let other people know what you've done. And if your supervisors, if your colleagues haven't told you yet, your research isn't actually complete until you've published it in some format. So why do we need to publish in English? Now, I know it may not seem fair if English isn't your first language, but the fact remains that English is the international language of science right now. Other researchers around the world, they know that good work is done here in Korea. And they want to hear from you. They want to know what's happening. By publishing in English, you can become a more effective communicator of science. Publishing in English gives you a wider audience. More people around the world will know what you're doing. Publishing in English also helps you to establish and enhance an international reputation. It also helps you with funding and will help you with academic promotion. Now, many researchers, especially younger researchers, find peer review to be a very negative process or a very difficult process to navigate. The fact is that peer review actually helps to improve your manuscript. There are very few papers that are accepted without any form of revision from peer reviewers. Rejection and revision are all integral and are all a part of the peer review process. And you should see the opportunity to revise your paper as a positive process. It will help make your manuscript better. But you need to be aware of what editors and reviewers want in a manuscript when you're submitting to a journal. Now, there are many, many things that journal editors and peer reviewers will look for in the manuscript that you submit to their journal. But there are two broad questions, two very important questions that they think about. They'll ask themselves, is the manuscript that's been submitted, is it sufficiently novel? And is the work presented in this manuscript, is it of broad enough interest to the people that read this particular journal? So you can see that reviewers and editors, when they're looking at a manuscript, they're weighing up the novelty and the significance of the work presented in the manuscript, and they're weighing it up against the aims and scope, the impact factor of the particular target journal. Now, over the years, both as a researcher and working as an editor for Edanz, I've had the opportunity to talk to many, many journal editors. And I asked them, what is it specifically that you're looking for in a manuscript? The number one answer that I get: journal editors want high quality research, the highest quality work possible for their journals. They want a manuscript that's going to stand up to the peer review process. They don't want to waste the time of their peer reviewers. Peer reviewers are a scarce, a precious resource because there are fewer people willing to act as peer reviewers than there are people submitting manuscripts to journals. So journal editors don't want to waste the time of peer reviewers by sending them poorly written manuscripts. Your manuscript, it needs to be original and novel in some way. It needs to advance your particular field in a small or a large way, but it needs to be novel. Journal editors only want work that will be of interest to people that read their journal. And it helps if you're working in an active research area, something where there are many citations being generated, many publications coming out in that field. Now, the second most common answer I got when asking journal editors what they look for in a manuscript, they want clear and concise English so that they can read the paper, they can read the manuscript quickly and make a decision quickly. So we can speed up the decision time, speed up the publication procedure. Now moving on to section 2, things that you need to think about before you start writing, and in a lot of cases before you start even doing any research. The first thing I want to talk about is reading. Now, we know that the human brain is made up of two hemispheres, left and the right side. One side's associated with logic, the other side's associated with creativity. Now, for us to function properly as human beings, we need both sides of the brain talking to one another. Left side and right side talking to one another, communicating. And the same can be said with research and reading and writing. To be the most effective researcher possible, you need to produce lots of publications. But at the same time, you need to read a lot. You need to be aware of the background literature and what's happening in your particular field. So reading and writing are very strongly connected processes. And you need to read as much as possible. The reason you need to read as much as possible, not only to keep up with the background material, but because reading improves your writing skills. So I've said here, read as often as possible. How often should that be? Well, you should be spending time every day looking for new articles, doing literature searches. Maybe 30 minutes every morning when you come in, looking for new abstracts. Then you need to dedicate time each week, maybe 1 or 2 hours on a Friday, and actually read the articles that you've downloaded during the week. Science is complex. Often the articles that are produced can be difficult to understand. So when you don't understand something that's been published, discuss with your colleagues. If you're not part of a journal club, if your department, if your lab doesn't have a journal club, consider starting a journal club where you can discuss these articles. Reading will help you with journal selection, finding the right journal to target. Because by reading a lot, you'll see where similar articles in the same field are getting published. Now, if they're publishing those articles, it's likely they're going to be interested in your work as well. And you can also get ideas for your next manuscript, for the next round of funding, what sort of grants you can apply for, what ideas to put into these grants. By keeping up with the reading, you identify gaps in the knowledge. It's likely that work needs to be done in this area. So you're getting ideas for your next manuscript, your next experimental study through reading. Now, there are ways to go about reading. You don't need to read an article from start to finish. You need to use your time effectively so that you can spend time writing, you can spend time doing work in the lab. And so this is a strategy for reading that I was taught when I was at university. That's also something that I taught to many of my PhD and master's students. on how to very quickly and effectively read published research articles. The first thing you'll always read will be the title and the abstract, because you're doing literature searches through databases. These are normally freely available. You'll be able to see these, and from there you make a decision on whether you actually want to download an article. The next thing you do is you need to self-assess how much do you know about a particular topic. Now, if you don't know much, or if you're new to a field, you've switched fields, it may be necessary to read the introduction of a published article. But if you're well into your PhD or your master's now, or you're experienced in a field, it's likely you've already read all the background material before. You've read introductions, they all seem to be very similar. There's no real reason that you need to read an entire introduction anymore. What you can do is go straight to the results, or the parts of the results that you need. Often this will be the figures and the tables. This is where most people will present their most significant results, what they think are their most important results. They know that people often don't want to read a large amount of text, so they'll present them in some sort of graphical format. Because that's how people present their work, that's how you should try and read it too. Go straight to these figures and tables. See what the authors have presented. The next thing you need to do is then read the discussion to see how they've interpreted their results. Do you agree, do you disagree with their interpretation? Only refer to the introduction or the materials and methods, the procedures section, if you need to. It's possible you may download an article purely for a particular method that's been mentioned in the paper, in which case you don't need to read any of the other sections. You can simply go to the methods. So think about strategies for reading. Think about what I've presented here when it comes to reading a published article. You need to be effective with using your time. Designing your experiments. It's a critical step to have good experimental design. So the question that you need to ask yourself, what is my hypothesis? What is my research question? And this will form the aim of your study, maybe for your entire PhD. What methods are appropriate? What techniques do I need to carry out? Do I have the resources? Do I have the skills? Do I have the knowledge? Will it be necessary for me to collaborate with another lab, either in Korea, somewhere else in the world? Do I have enough money to carry out this work? Things that you need to think about before you start doing any work. Identifying your controls, positive and negative controls. It's important to do this and to explicitly state them in your manuscript, because this is something that peer reviewers look for. Straight away, peer reviewers will go to a method section, see that you have the appropriate positive and negative controls. A lot of science involves gathering large amounts of data and performing statistical analysis on this to see whether it's significant, whether the changes observed are statistically significant. So, have you collected enough data? Your sample sizes, your populations, are they large enough? Which is the right test? There are many, many tests, statistical tests, that are available for use. Which is the right one? Depends on how you've sampled your data. Now, when I was doing my PhD, when I was a postdoctoral researcher, statistics was very difficult for me. It still is very difficult. So often when I was designing my studies, and while I was carrying out experiments, and afterwards when I was analysing, talk to a statistician to get the best advice. I talked to them and said, this is what I want to do, this is what I'm hoping to show. What's the best way that I should go about doing this? Statistics is a branch of maths, a branch of science by itself. It's very complicated, so it's unlikely that you're going to be an expert in statistics as well. So talk to the experts, talk to the statisticians for advice. The study that you're going to carry out, have you fulfilled all the ethical requirements? All ethics requirements need to be fulfilled before you start doing any work. So if you're doing a clinical study, do you have patient consent forms? Clinical trials, have they been registered? Animal experiments, genetically modified organisms, materials transfer agreements, they all need to be approved before you start doing any work. And now more and more journals are asking for proof that these sorts of ethical requirements have been met. So I want to talk a little bit about journal selection now. So how many people in the audience, show me your hands, struggle to identify a good target journal? They're confused when it comes to selecting a journal to submit their manuscript. Who finds it difficult sometimes? Okay, so I see a few hands there. On this slide, this is just a very, very small selection of journals, peer-reviewed journals available for publication. There are more than 20,000 peer-reviewed journals in the world for you to submit to. So which is the right journal? Which is the most appropriate journal to send your manuscript into? Well, the decision about when to decide on your target journal is an important one to think about. Now, many people will write their manuscript, they'll have a full draft of their manuscript, and then decide on a target journal. That's completely the wrong way to go about selecting a target journal. It's something you need to be always thinking about. So, as you're doing your work, you're always collecting your results, and you're analysing your results as you go. At some point, you make a decision, I have enough results for a paper. And at that point, you should be focusing your thoughts about a target journal. You need to make a decision. How high to aim? Is your work of high significance? Is it of low significance? And I'll talk a little bit more about significance and how to evaluate the significance of your manuscript. Definitely, before you write the title, the abstract, introduction, or the discussion sections of your manuscript, you should have one single target journal in mind that you wish to submit to. You need to match your manuscript to your particular target journal. This is why you can't write an entire manuscript and then try and target it towards a journal. You need to think about it as you're writing. So what is your message in your manuscript? What idea, what concepts, what information are you trying to get across to the readers? Now, when I talk about readers, I don't mean the people, or just the people at the end that will download the published article. I mean the journal editors, I mean the peer reviewers. These are readers as well. Who will be interested? So, the readership, the target audience. Will it be just for clinical immunologists? Or will it be for all people in the life sciences? Think about this. Just theoretical physicists. Think about your target audience. I said again, the significance of your results. How significant are they? Highly significant? Or of a lower impact? And where have similar articles been published previously? Well, how do you know that? It goes back to your reading. Doing as much reading as possible. Now, there are lots of factors to consider when it comes to selecting your target journal. I've listed just some of them here, but it depends on your publication goals. What you want to achieve, how quickly you want to achieve it. So, for some of you, there may be a pressure to publish at a certain impact factor. Maybe your funding requires you to publish in open access only, because it's publicly funded, therefore the general public should have free access to that work. When I was publishing as a student, as a researcher, the most important thing to me was the target audience. I wanted the right people reading my published work, because if the right people are reading it, then it's more likely they're going to cite it as well. That's something else you need to think about, citations. Being an academic, being a researcher is not just about having lots of published articles. it's about having people cite your work as well, because that establishes your reputation. It makes, the more you're cited, the more you're seen as an expert in your field. So, which factor or which factors are the most important to you? Could be a combination of some of these, maybe something I've not even listed there today. So you need to think about these. Now I said we'd talk about evaluating significance. You need to think about your manuscript, you need to assess it. You need to be honest when it comes to this assessment. So, the work, is it of specific interest presented to for only a few people, or will your results, will they be of interest to many, many people? Will it affect a lot of people? Is it a new tool, a new treatment? Does your work support or contradict an existing theory? Does it improve our understanding of a particular phenomenon? Or does it provide us with some new technology that can be used globally? If it's the case, if the answer to any of these is yes, it's interest to many, or affects a lot of people, it's likely that the significance of your work is increased, the significance of your manuscript is increased. The novelty of your work, how new is your work compared to what's previously been published? Now, by definition, journal editors, your work should be novel in some way. So you have new findings of some sort. But it's an incremental advance, it's a small stepwise advance in the global knowledge base. Well, honestly, it's probably going to be accepted by low to medium impact factor journals. But if you've made some sort of conceptual advance in a particular field, then it's more likely that medium to higher impact factor journals will be interested. So you need to be aware of how new your results are compared with others that are already published. Now, when I talk about impact factors, we need to remember that different fields have different scales for impact factors. So, from the impact factors that were published yesterday, we can see that the highest impact factor journal for physics is around 2.216. Whereas in clinical medicine, the highest impact factor journals available reach into the 20s or the 30s. So in virology, when I used to publish in that area, the highest impact factor journal in virology was around 5. So when I'm talking about high and low impact factors, it depends on the field that you're working in. The relevance of your work. So your results, are they only specific for a particular geographical region or a particular ethnic population? Or does it have worldwide implications? Higher impact factor journals will consider very specific findings confined to a particular area if they're the very first of their kind or of international significance. So it's the very first time something's been reported, even if it is in a regional area. The higher impact journals may be interested in it. The appeal of your manuscript. Is there popular appeal? Is this subject matter being reported in the mainstream media? If it is, then it's likely that the significance of your work, of your results, are increased. Now there are certain hot areas of research. These are just some current examples. Optogenetics. Global warming, because it affects the entire planet. Stem cells and their use because it's very controversial. It's always in the news. Anything related to the Higgs boson particle or involved with the Large Hadron Collider. Working in these areas or trying to tie your work into these areas increases its significance. Now, I don't want to talk too much about publication ethics because I think JB has covered it in quite a lot of detail. And I think he did a very good job of it. He's mentioned almost all of these points here. The point I want to point out though is plagiarism. the idea of plagiarism. There are lots of problems with plagiarism around the world. It doesn't matter if it was done on purpose, if it was accidental. It's still considered plagiarism by reviewers, by editors, by the scientific community. And plagiarism has significant consequences, not only on you as an author, but your co-authors, the institute that you work at, and in some cases even your entire country. It can really tarnish the reputation of a country if lots of its researchers are plagiarizing. I've mentioned the improper use of human subjects and animals. Hopefully that won't happen to you, because in the study design phase, you've already obtained all the various ethical requirements that you need. All the paperwork has been filled in. Something that is becoming more and more common, confusion relating to conflicts of interest, or potential conflicts of interest. Where funding has come from, when you know people at funding bodies or you're related to somebody somewhere, there can be a potential conflict of interest. Editors, reviewers, everyone is aware that these potential conflicts of interest exist. And you need to clearly state this when you submit a manuscript, if there is a potential conflict of interest. By doing that, it tells the editor, it tells the peer reviewers that even though I was funded by a particular private company, or I know a person on this particular funding body, I've still tried to do my work as objectively as possible and present it as objectively as possible. There aren't any standardized international guidelines as yet, but there are many guidelines that are recommended by various scientific bodies. Now, before I go any further, I think we're doing quite well for time here today. Are there any questions on anything I've presented as yet? Maybe something on publication ethics that JB didn't cover. Something on the publication process. Timing. No, if we've got no more questions, then we'll move on. We'll move on to section 3. How to go about structuring a manuscript. So when I was writing up my honours thesis, when I was writing up my PhD thesis, my supervisor always used to say to me, "You're always telling a story. You've always got to tell a story in your thesis, in your manuscripts." And we say in English, every great story has a beginning, it has a middle, and it has an end. And your story about your research needs to be easy to read and easy to understand. And these are two different things that I'll talk about in these next 2 sections. There's an approach to writing, the tell them 3 times approach that I was taught in high school. So we have the beginning of our story. It corresponds to the introduction of our journal manuscript, of our book. And it's here you make an assertion, or a number of assertions. And you tell the reader what you're going to tell them in the rest of your story. Then we have the middle of our story, it corresponds to the main body, the results section, maybe even part of the discussion. That's the evidence. We say, in the body, in the middle of the story, we tell the reader. We tell them the information. Then we have the end of our story, our conclusion. And it's here where we provide some sort of affirmation of the work we've presented. And so we tell them again, we tell the reader again, what you've told them previously, at the beginning and the middle. That's what we call the tell them 3 times approach when it comes to writing. Now, the majority of scientific manuscripts are broken down into discrete sections. And it follows, in general, something called the IMRAD order. And IMRAD stands for Introduction, Methods, Results, and Discussion. That's IMRAD. Now, different journals, different fields will use different headings. Methods may be called procedures, or materials and methods, or techniques. Sometimes results and discussion sections will be combined. Sometimes the methods will be presented after the discussion. Depends on your field, depends on your target journal. but almost every scientific manuscript published follows or contains these 4 sections and some sort of abstracted summary at the start. The introduction is where we have our assertion, the results are where we present evidence, and the affirmation comes in our discussion section. However, there's a problem with the way articles are presented, because people see articles presented in a certain order and they try and write them in that order. Well, that makes it difficult for the writer. So there is a better way to write. For ease, for clarity, for consistency. While you're conducting your research, you can write your methods and your results. You're always collecting results. You're always analysing them, so you can start writing them as you're doing your research. Many times you can actually start writing your methods before you start doing the research. You know which methods you want to use. You can start making notes about these. Once you've collected all your results, you've made your decision you want to publish, you then need to have your one target journal selected. Once you've selected that target journal, you can start writing the introduction and the discussion, following the target journal's guidelines. The very last sections you should write, even though they're presented first when they're published, the title and the abstract, because they're a full summary of your entire manuscript. And you can't write an effective title or an effective abstract until you have a full draft of all of these sections. So let's look at all the different sections of a manuscript. The first section, the abstract. Abstracts need to be concise. Once again, depends on your target journal. Maybe 100 to 300 words. It may be shorter, it may be longer. The first thing you need to present, you need to describe the problems addressed. What were your objectives? What were your hypotheses? Then tell the reader in the abstract what techniques you used, what methods you used. But this only has to be very general. The common mistake I see every day when editing manuscripts from authors all over the world is that they provide a lot of detail about the methods and techniques they used. And then there are hardly any results presented. This is the section in your abstract where you need to provide the most amount of information. This is what people want from the abstract. This is what the reader wants. Think about your reader. Make it easy for them to understand what you found. Present results in more detail than you provide your techniques. You don't have to present all the results, just the ones you think are the most important. The very last thing in your abstract should be your concluding statement. The major conclusion or conclusions of your work. Now let's move on to our introduction. The question that you need to pose, what problem was studied? That's the question you need to think of. And the answer to this question should be in the introduction. Now, I've talked about telling a story and how your entire manuscript needs to have the beginning, middle, and end. Well, to make it easy for you as a writer, think about your introduction as having 3 parts as well, as having a beginning, a middle, and an end. Let's look at the beginning of our introduction. So you need to provide sufficient background information. Now, the way to provide this information and put your work into context is to start very broadly. Start general and become specific. Avoid what I see almost every day, another mistake I see. People tend to write very, very long introductions. They tend to be comprehensive literature reviews. There's no need for that. What you can do is cite the reviews, cite review articles, point the reader in the right direction, tell them where they can get information if they need it, if they want it. Then in the middle of our introduction, we need to provide the rationale, the reasons for doing this work. Why was it important that we do this work? Justify your work to the readers, to the peer reviewers, to the editors. And explain to them how you tried to address the problem. Again, you're avoiding details. Make some generalized statement about the type of study you did, the type of simulation you conducted. Again, what you should avoid doing, don't state the results in the introduction. I see this very often. Save the results for the results section. The introduction is simply to provide a background to the reader, for the reader. Then we come to the end of our introduction. Our last 1 or 2 paragraphs of the introduction. Here, state the very specific methods you plan to use. Again, no need to go into any great detail. Then very clearly and explicitly state what the specific aims or objectives of your work were. I'm always surprised at how many authors don't do this in manuscripts that I get before submission. If you don't state what your aims and objectives are, they often confuse the reader. So make it very obvious. It's also important to do this in the last paragraph, because when people are taught to read, so if you think back to the reading strategies slide, when people are taught to read an introduction, if they already know the background material, they'll often just go to that last paragraph to find out what your specific aims were. That's where they believe that information should be. So it's about making it easy for the reader to understand. Our methods. If your journal allows it, your target journal, use subheadings. Present them in a logical order. Often present them in the order that you conducted the work. Use the past tense. If you've developed a new method, you need to describe it in enough detail so that another scientist, another researcher can repeat your work. But when you have an established method, there's no need for you to describe it. Simply refer back to the previous publication. Save yourself time, save yourself effort. You don't need to try and describe it again in your own words. If the reader wants details about that method that you've used, they can go into the citation that you've provided. Okay, results. What you found. Again, use subheadings and try and use similar or the same subheadings that you use in your methods section, and try to present them in almost the same order as well, because that's a logical flow. It's what readers are expecting as well. It helps them with understanding your manuscript. Again, use the past tense because it's work that you've conducted, it's the findings that you found in the past. But when referring to figures and tables, we use the present tense. Again, things that you shouldn't do. Don't try and explain your results. Don't try and provide what the implications are. That comes later in the next section. Avoid duplicating data in figures and tables and the text. Only present it in one way. Now, the great thing about display items, about figures, tables, schematics, maps, they allow you to present a lot of information very quickly, very efficiently. Now, remember what I told you about reading strategies. When you're reading something, you have the knowledge, you can go straight to the results. Look at the tables and the figures first, because that's where you should be presenting the most significant result, as some sort of a figure or table, if possible. But for a figure to be effective, to convey your message, it needs to be simple as well. So if you have a lot of data to present, try to break it up into separate panels, if possible. I mentioned duplication on the previous slide. So when you present a figure or a table, don't try and explain it in the text. Simply refer the reader to the figure or the table. Label all parts of your figures. And the legends, the captions to your figures and tables, they need to be standalone. So when I go to a published article, I see a figure or a table. I want to be able to look at this figure or table and understand the message that the author is trying to tell me. I shouldn't have to go back to the main manuscript. and get information from the manuscript to understand the figure or table. All the information that somebody needs to read should be in that figure caption, in that table legend. So this is an example of what I think is a very good table, the way that data is presented. The authors have provided a very clear and concise legend that tells me all the information I need to understand what's presented here. The authors have really thought about how to format this table. They've thought about spacing, presenting it in columns that are easy to differentiate. There's a nice gap between each column. To save on space, they've used these particular abbreviations. For me to understand what these abbreviations mean, though, there needs to be some sort of explanation, so they've defined them as a footnote to the table. So I'm sure in this particular manuscript, these definitions were in the manuscript, but to avoid me having to go back there through the text and try and remember what the abbreviations were, they've defined it for me. An example of a good figure. You can see it's been broken up into 3 panels, A, B, and C, and we have an inset here as well. See, there's a very detailed legend. It's standalone. It tells me everything I need to know. You can see different colours have been used. What do they mean? What do they correspond to? The axes for each graph have been clearly labelled. See, in these fluorescent images, we've got scale bars. So we know the size of what we're looking at. This is considered an effective figure. It conveys a lot of information in a very simple and easy to understand way. Now let's move on to our discussion. What do your findings mean? So this is the question you ask yourself about your discussion. What do my findings mean? And the answer to that question is contained within this section. So like our manuscript, our entire manuscript with beginning, middle and end, we can think about our discussion as having a beginning, a middle and an end. And think about what information goes in each part. So at the beginning of our discussion, avoid just copy and pasting, avoid repeating your entire results section. What you need to do is make some sort of generalized statement about your most important result. You posed a particular research question, there was a problem in the introduction. This is where you get to answer that question for the reader. Emphasize what you think is the most important finding first of the work in your manuscript. Which is the most important? This is what you should state in the discussion. Based on this major finding or findings, what are your major conclusions? This all should be said in the first paragraph to your discussion. This is what readers are expecting, so you should provide it for them. In the middle, this is where we do interpretation of all our results. So, our most important result that we presented first, we explain it, and then all the other results that we presented in the previous section. So you need to do comparisons with other studies that have been published. Are your results the same? Are your results different? What are the reasons for this? Why are they the same? Why are they different? Did you get the same results, but achieve it in a different way to what's been published previously? That's important. If your results differ, why do they differ? Previous publications, are they right? Are they wrong? If you've presented any unexpected results in the previous section, you need to try and explain them in the discussion. Peer reviewers expect it when they're assessing your manuscript. If you've presented a result, you need to explain it in this section. If you can't explain it, if it's too difficult, there's no previously published material, then you need to seriously reconsider about whether you can include that result in your manuscript. Describe the weaknesses, the limitations of your work. Now, this may sound odd to a lot of people, but It's important that you do this because it shows the reviewers that you're thinking at a very high level. You need to tell the reader how your experiments could be improved. What were the flaws? Reviewers really like it when researchers do this. You're providing advice for your fellow scientists when this manuscript gets eventually published and is available for other researchers to read. You're telling them what not to do, as well as what you found. So you're helping the scientific community. At the end of the discussion, restate your major conclusions. There are these, what we call signposts in English. So you can start a sentence with, in summary or in conclusion, we found this, we found that. These are very powerful statements to make. And a common mistake that I see in manuscripts every day is people using these throughout their entire manuscript. In the introduction section, in the results section,